So we have previously seen that a range of cloning vectors are present, and the selection of those vectors primarily depends on the what is the purposes of using that vector, or the downstream processing, how the experiments has been done, and what is the goal of the experiment. So in this case, there are other factors we need to be considered for choosing a cloning vector. And firstly, the insert size, as we have seen that the bacteriophage, which cannot be taken in long fragments. On the other hand, the back or the yak, they can take a long fragments, and they are very suitable for sequencing the mammalian genomes. Copy number, the next factor is that how many copies you want at the end of the transformation. Do you need a lot of copy numbers to be present in your transformant, or is a low copy number is fine? In that case, we need to select those specific vector and a specific host cell, or that is bacteria. Incompatibility, sometimes there need to be in a transform two plasmids into the same host organism. And in this case, the two plasmid, if they have the same origin of replication, they can compete each other for the replication machinery, and they are unable to coexist at the same host. So we need to be careful about this as well. Selectable markers. Normally, antibiotic resistance has been used as a selectable marker, but in other cases, there are a lot of other selectable markers can be used, like a growth in a specific nutrient. So this can be done depending on how your experiments has been designed. Then the cloning site, there are a lot of there is a multiple cloning sites, but you need to be make sure that the insert is restriction, the digested product of the insert is actually compatible with your vector. So the vector has the same restriction digestion sites that can be ligated to the insert can be ligated to the cloning vector. And also the specialized plasmid functions like the expression vectors. As I said, that this is very important for the biotech industries, because it's mostly used for the protein production. And for the protein production, it needs to have the 5' promoter, the translation sequence, the termination sequence, everything. The second point of the molecular cloning is the preparation of the vectors and the DNA. So it means that we need to make it ready for the vector and the DNA so that they can be ligated as a one product. So in most cases, restriction digestion has been used for this purpose. So restriction digestion is one of the widely used techniques in molecular cloning. Restriction enzymes work as a molecular scissor. For more than 50 years, restriction enzymes remain an essential tool for recombinant DNA technology. The restriction enzymes were identified as a defense mechanism of bacteria against the bacteriophage. Most of the restriction enzymes have related DNA methylases that recognize the same sequence as restriction enzymes. The methylases transfer a methyl to the cytosine and protect the DNA from degradation, which we will discuss a bit later. Generally, restriction enzyme recognizes a specific sequences of 4 to 6 base pairs and then cut the both strand, either making a sticky ends or a blunt end. Here we can see that the restriction enzyme EcoR1 recognize the sequence 5' GAA TTC, that's 3', and binds as a homodimer and cleave the sequence between the G and A from the 5' end, and creating a fragment of DNA that carry an overhang of single-stranded DNA. This kind of single-stranded termini, also called a sticky end. On the other hand, restriction enzyme SMAB1 that recognizes the sequence from 5' to 3', C, 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 G, G, G. And it binds as a homodimer and cleaves between the C and G. However, SMAB1 cleaves both strands exactly at the same place and generate a blunt end DNA fragment. This is a blunt end DNA fragment. There is no overhanging single-stranded DNA as we have seen in EcoR1. As you might notice that both enzymes recognize a sequence that read same from the 5' to 3' end. So from 5' to 3' end, it is the same sequence. And these kind of sequences are called a palindromic sequence. Restriction enzymes broadly classified into three major groups. So the type 1 restriction enzymes, that is cleaved randomly about a 1000 BP away from the recognition site. So it is not really cutting the same recognition sequence. The recognition sequence has been identified and it has been cleaved the sequence randomly non-specific sequence, which is at least a 1000 base pairs away from that recognition site. It's contain the endonucleases and the methylases in a single protein molecule, as we have discussed previously. That the methylases is protect the DNA and endonucleases, which is cleave the DNA. The type 2 restriction enzymes, which are mainly used in the laboratory, they recognize and cleave the specific sequence. So like the example we have given for the EcoR1 and SMAB1 in our previous slides. And their endonucleases and the methylases are in separate protein. The type 3, like the type 1 restriction enzyme, they also cleave a non-specific sequence, which is around 24 to 26 base pair away from the recognition site. And their endonucleases and methylases also located on a single protein molecule. As we just discussed a few slides back, that the restriction enzymes also have another component in their system, and that is the methylases. So the methylases, what they do, they transfer a methyl group, especially to the cytosine residue, and form the 5' methyl cytosine. And that methyl DNA is particular sequence to prevent from being degraded by endo restriction enzymes or other foreign bodies. A prokaryotic actually have a gene encoding one or few restriction modification systems containing the methylases. And this, as I said, that this is adding the methyl group to the cytosine molecule. And on the other hand, its companion, that is the endonucleases, which recognize the same sequence and which cleave the DNA, which is not methylated. This diagram is showing when the EcoR1 restriction enzyme, it is cutting the DNA or cleaving the DNA in the recognition sequence. However, when this same sequence has been methylated, which is the methyl group has been transferred by the EcoR1 methylases. So when the same group has been methylated, the restriction enzyme cannot cleave that same sequence.